I'm very excited to be able to chat with you all today in the CAIRIBU group. The part of the talk that's about science will focus on this polymer called chitosan and how it affects the urothelium. But with this, with the K12 program being a career development program, the other half of the talk will kind of focus on this process of finding a research niche and setting all the plans in place for a career in academic science. So disclosures, as Alex mentioned, I am co-founder of a company that I'll talk about briefly that's related to some of the research topics I'm involved with. And then also I'm a urologic oncologist, but the focus of this research is benign. So for context, this has been my path as an MD who did not do a PhD. And it was kind of interesting to reflect on a relatively fragmented pathway. So I think it was good to have a very strong scientific foundation from Caltech as an undergraduate. But then during medical training, there's just years at a time where you don't really have any time to think about science. Then you come back to the lab for a year, then go back to clinical and back and forth. And I'll just tell you that at the start of this K12 year, I was really overwhelmed by all the new advancements in science. Like RNA sequencing wasn't even a thing the last time I studied molecular biology. But my goal for this year was to be able to convince someone to let me do science and surgery for a job, and I needed a research vision to show them. So luckily, I was successful. I'll be starting out my own lab at Stanford in the fall. Being on staff as a urologic oncologist, but developing that scientific vision and strategy has been a really interesting process that I'd love to share with you all alongside discussing my current research on urothelial exfoliation. So just first, there's one resource that I like to foist upon everybody I come across. This was part of a class that I took during a HHMI Medical Fellows year on this book called Writing from the Reader's Perspective. And I've heard this a few times throughout CAIRIBU talks about writing from the grant reviewer's perspective, et cetera. So I just wanted to share this link. That's a really nice exercise about getting into the science of how to structure your writing down to the clause of the sentence. So just throwing that out there. All right, I'm going to give a couple of context slides about how I ended up researching chitosan, as otherwise it seems kind of random. But back in 2015, as a medical student, I worked on this really interesting project to try and get more circulating tumor cells in a liquid blood biopsy. So, for example, even in metastatic disease, the number of cells that you might find in a blood sample of circulating tumor cells is really, really low. And so the utility of this test was pretty limited. It would help in prognostics, but you couldn't, test your personalized therapies and things like that. So we use the concept of magnetic separation, which is using magnetic nanoparticles that have been tagged with an antibody against your cell or target of interest. And then we designed this tiny magnetic wire that could fit through an IV to sit in the vein. So the idea is you infuse the magnetic nanoparticles, label the cells, then use your magnetic wire to fish them out and get a much higher yield that you can then use for cell culture, and much more personalized studies. So this was during medical school, research year ended. I went back to medical training for several years and started residency. And then during residency, started doing a lot of ureteroscopies. And so for non-urologists in the room, the most common surgery we do to treat kidney stones is via this ureteroscope, where we put a small flexible instrument through the urethra, up the ureter, find the stone, break it apart with a laser, and then try to take out all the pieces that we made so that they don't just grow back into bigger stones or reobstruct and cause pain again. Unfortunately, for decades, we've pretty much had one type of tool to do this with, which is this wire basket. And it's really, really tedious. You can be there for hours, and half of our surgeries don't actually get the patient stone free. So now, as the junior resident who's basketing stone fragments for hours on end and pretty miserable, I wanted to apply that magnetic retrieval technology to kidney stones, thinking, hey, there's just got to be a better way to do this. Um, and in some ways, for stones, it is the same principle. You coat your target or the kidney stone fragments with magnetic nanoparticles that stick to the stone and pull them out with the magnetic wire. So for stones, you don't need a specific antibody. You just need a negative charge because the surface of your kidney stone crystal has these positively charged exposed calcium ions, and then they all stick together. However, you'll notice from the scale bar here that this is really only capturing submillimeter dust fragments, which is not really good enough for clinical use. And the reasoning behind this is pretty simple. It's just kidney stones are a lot heavier than cells, so you need a lot more particles per target and able to be able to actually magnetically retrieve them because we kind of maxed out how strong our wire can get for that given size. And so here's finally where chitosan comes into play, is we made a magnetic hydrogel. And so this is done with ionic gelation. Our particles are negative, so they stick to the stone. Then we add a positively charged polymer that glues them all together. And so on the right-hand side, you can see that with just iron oxide nanoparticles or these spions, you basically just have a monolayer. You can't really see them on top of this jagged crystal surface. But with the hydrogel, it's many microns thick. You're really amplifying the magnetic moment that you're generating. And so with the chitosan hydrogel, we were able to retrieve fragments that were much larger in size, so not just that submillimeter dust. It worked for all different kinds of kidney stone compositions because it's just all stones are ionic crystals. And this research has led us on a really interesting journey in terms of translation, because I think everybody's goal is to be able to see the research that they do in the lab eventually be translated into the clinic, be used in patients, and I think we're on that track, which is really cool. So this video that I'm showing is the actual device that would be used to feed it through the ureteroscope, be able to inject the gel under direct vision, and then passing the magnetic wire in to retrieve it. So this has involved a lot of funding and a lot of very tiny tubes. But recently, this has been spun out of Stanford into a company, and it's been really illuminating to see all the other additional work that goes in to actually get the science from the lab to something that could is a real product and could help and treat patients. So it's also helped me get a realistic understanding of how that actually happens in this country, which is effectively it's going to cost way more money than you can get from grant funding. So you need to be able to convince investors that you have the foundations of a business instead of just a science project. And it's really fundamentally different from how we're taught to think in academia. I don't really like it, but it's been really useful to understand and be able to anticipate the types of questions that you will get from investors and FDA so that you can make sure that you're setting yourself up for success. So this is kind of a common mantra in the biodesign role, which is starting with the end in mind. And so back onto the science part of it. So besides make hydrogels, what else does chitosan do, and what is chitosan? So it is a positively charged polymer. It's derived from chitin, which is found in the exoskeletons of crustaceans like crab and shrimp, also in fungal cell walls. And so it's a really abundant biomolecule, meaning that hopefully for translational purposes, it can lead to some very cheap drugs. We'll see about that because again, with the conflicting interests, but theoretically we could have very cheap therapies made from this molecule. There's a lot of other really cool things about chitosan. Every description of it, for the most part, has mentioned its safety, biocompatibility. FDA has recognized it as a generally safe material to put in food and drink. So some of the example applications are its antimicrobial properties. So all of these really stem from how positively charged this polymer is. So bacteria have negatively charged cell walls. You coat them with chitosan, it kind of smothers and kills them. And so actually in our meat packaging, chitosan is really prevalent to help prevent spoiling. If you like wine, you got to get all the impurities out of your wine. So people use chitosan for that. It's also used in wastewater treatment plants. Clinically, it's FDA approved as hemostatic agents because it binds together all the negatively charged platelets and red blood cells. So this is actually, it's used in trauma settings because it's so good at stopping even arterial bleeds. And then finally, our mucosal membranes, such as what we have in the bladder, are lined with these negatively charged glycosaminoglycans. And so chitosan really sticks to that as well and is used to improve drug delivery in a variety of applications. Furthermore, we even see suggestions that it has, benefits in wound healing. So this is a paper where they inflicted a skin injury on a rat and treated one group with chitosan, and it's really sped up the healing. And we also see chitosan in FDA-approved wound dressings. So this is where things get a little bit more mysterious because maybe the wound healing is just because it's got antimicrobial and hemostatic properties, which can be explained by the charge, but it's also shown to reduce the amount of fibrosis and have an anti-inflammatory effect. And so there's likely more to the story in terms of mechanism. And now, for all these really kind of fascinating applications, what does it do in the bladder? It actually has a really dramatic and unique to the urothelium effect in that you put a solution of this in the bladder and it causes the entire urothelium to just slough off in a dose and time dependent manner. So when we were studying it for the magnetic hydrogel for kidney stones, it was all bound up in the hydrogel form, so it's not too active. But we were looking at it by itself on ex vivo urothelium and saw that if you increase the concentration and duration, you can get this full thickness exfoliation. However, it also regenerates really, really, really quickly. So these are images of mouse bladders where we've stained the superficial urothelial cells in red and the basal ones in green. And you can see that with even just 15 to 30 minutes of exposure, you've completely gotten rid of the urothelium. But by 12 hours, you see the green basal cells starting to regenerate, and it looks pretty dang normal at 48 hours. And so, as I was thinking, working on this project during residency, knowing fellowship and the rest of my career were coming up, and wanting to find an independent research focus, this topic seemed really meaty and something that could sustain multiple lines of research. For example, studying why chitosan does this. How can we use this as a research tool to have a really controlled and tunable way to induce urothelial exfoliation and regeneration compared with other models that we use for bladder injury like infection or cyclophosphamide, which are kind of generally a lot more injurious in terms of magnitude and a little bit more difficult to control. If it's got these magical wound healing properties, can we apply it urologically and help injured urothelium regenerate better? If it's too diseased, can we just get rid of it? And what information can we get from these exfoliated cells to help with our diagnostics? And so envisioning how these tools would actually look like and be used in the clinic has helped shape the overall strategy and help think about the regulatory pathway of eventually getting this out of the lab. So as I was digging into this, call back to yesterday's talk on writing specific aims and such, I'm aware that this is not a totally new finding. There's a nice series of papers that are already discussing chitosan-induced exfoliation. And even back in 1988, somebody put chitosan in a dog and showed that it helped heal urothelial injuries. But, if chitosan is so safe and wonderful, why don't we have a product on the market for our patients yet that's taking advantage of all of these effects? And again, looking into the data that's out there, you also look into the gaps and controversies that surround them. And I would say there's quite a lot for chitosan. The main barrier, I think, is that people one, people don't really understand how it works, but that's true for a lot of the therapies that we've managed to translate into the clinic. But I think the trouble is that so far people for, have not been able to really characterize or know what they're even testing. So chitosan's a polymer with a bunch of variable properties, molecular weight, degree of decetylation, and therefore its charge, etc. And it's not really been consistently reported between studies. So we have a dozen papers saying chitosan's pro-inflammatory, another dozen saying it's anti-inflammatory. And oh, by the way, maybe everyone's chitosan is just super contaminated with endotoxin, and the endotoxin is why you're seeing all of these weird immunomodulatory effects. And so, thinking forward to potential translation, FDA is going to have a lot of questions, and if we can't trust the evidence behind what we're saying, then it's just not going to move forward. So my goal for this overarching research program has been to establish a really rigorous and well-characterized set of studies to answer the specific question of what is it doing in the urothelium, hopefully figure out how it's doing it, and then testing the hypothesis behind our proposed clinical applications to use it to help improve urothelial healing, among other things. And what the K12 has been really important for is figuring out how I am going to accomplish this as an independent investigator. So shout out to our K12 PIs at Stanford, Dr. Joseph Liao and Jim Brooks, and then our other scholars, Alex Velasco and Katie Kahn as well. So as I mentioned, chitosan's not just one molecule. It's a polymer, so it can vary a lot in molecular weight, which affects its viscosity on the horizontal axis here, depending on how it was processed to deacetylate it and turn it from chitin into chitosan. It can have a wide range in its positive charge on the vertical axis. And you can buy chitosan from a lot of different places. So each of these bubbles is a type of chitosan that we were able to purchase from a manufacturer. And so you can see there's a huge spectrum. There's also, is it soluble in acid? Is it soluble in water? And so already you can kind of see what a problem we have in the literature, because most people just say we use chitosan, right? And so kind of one of the first things we set off to do was to figure, okay, is there a difference between all of these? And to make a pretty long story relatively short, for the purpose of exfoliation, they all seemed about the same. And so on the bottom, you see some H&E slides of mouse bladders, where these little stringy bits are actually sheets of superficial umbrella cells that are peeling off with the chitosan. And then actually the leftover chitosan stains really pink because the eosin in H&E is really negatively charged. And so that's kind of a cool finding. But to summarize, I didn't see a huge difference between all of these. I still don't think I have the optimal assay for this. Cell culture wasn't going to work because we needed to study a multi-layered urothelium. In vivo bladders are a little difficult because it's hard to distend them all perfectly the same way. Sometimes they leak, sometimes they don't. But at this point, it felt relatively comfortable just picking one and moving forward to analyze that specific one in depth further to look at the mechanism. And now for the mechanism of how chitosan causes urothelial exfoliation, I have two main hypotheses. One is more physical based. And so if we go off the positive charge being responsible for most of its known effects, we can think about what happens when chitosan binds to the negatively charged cell surface proteins. So this top study looked at chitosan in intestinal epithelial cells, and it shows that it actually physically clumps together those integrins and disrupts your tight junction proteins. So with that, we know that it really increases the permeability of the epithelium, and so it's used a lot in drug delivery. But then if you think about all the consequences of disrupting your tight junctions, there's also, plausible pathways for how this triggers cell death pathways like caspase proteins for apoptosis. And so that's one theory that the mechanical disruption is leading to cell death. However, this pattern of response is also pretty similar to what we see in UTIs. So exfoliation is the bladder's defense mechanism to bacteria. This top study came out of Duke, and it showed that with exposure to uropathogenic E. coli, within 12 hours, again, this superficial layer in green in these pictures is widely exfoliated. And they dug into the mechanism more and found that the bacteria activate the inflammasome that releases cytokines attracting mast cells and then leads to pyroptosis, another mechanism of cell death. So it's also possible that this is triggering a similar immune response to cause this really rapid exfoliation. If the cells are coated in chitosan, does the bladder just think it's got bacteria stuck to it and triggers the same pathways? So we're going about this in a few different ways for approach, but one is to do bulk RNA sequencing experiments to help cast a wider net than just these two hypotheses. And also just thinking forward, I knew I needed to gain some understanding of this to be able to keep up with the modern literature. So thank you to Dylan, who's one of the Stanford residents with bioinformatics experience, who's been helping me out. And then also Sarah Doe is a really amazing undergraduate at UCSD. I had the pleasure of mentoring her through our pre-renal summer program, which again is just the cutest name. But she's continuing to work with me to help on this data analysis. So we performed intravesical installations in mice with either just water as a control or a chosen chitosan formulation, and then also LPS or endotoxin as a comparator. So you'll remember one of the questions in all this is, well, is chitosan's activity just from endotoxin contamination? We've tested what we have, and it's actually got pretty low levels, which is reassuring. And then for these mice, we took the bladders at 4 and 24 hours and looked at the bulk RNA sequencing data. So we're still parsing through most of it, but to try and answer one of the specific questions of is there a significant immune response related to chitosan-mediated injury, the answer seems to be no. So the immune microenvironment of the control water installation and chitosan at 4 hours, the left two bars, are pretty similar, while the endotoxin exposure group has a significant neutrophilic infiltrate at 4 hours. And so we're also reassured that they're doing two different things, and chitosan does have some effect that's unique of just endotoxin. And so there's still a lot of work in progress on the mechanistic evaluation. I wanted to just briefly talk about the plans for the second aim, which is to understand how the urothelium regenerates after chitosan injury. So we've seen it basically regrow within a day or two, and then the other wound healing data from the skin studies suggests that it also leads to much better wound repair. And so I plan to compare this with our existing models of bladder injury, for example, cyclophosphamide, which we know can cause hemorrhagic cystitis and is used to model overactive bladder in mice because it leads to this chronic inflammation. And so we'll plan to compare the two models with the similar methods that we described before and see what differences exist in gene expression as the urothelium heals. And then finally, if chitosan does recruit some sort of friendlier repair response, what happens if we treat injured urothelium with chitosan? Can it override that signaling that leads to chronic injury and kind of do a factory reset of the bladder and hopefully support its translation to therapeutic applications? So I am going to close out by summarizing some of the pitfalls I've been trying to dodge with various levels of success. But the first is to really know what molecule you're working for. And I think this is applies for anything that you want to translate out because you have to know how it's made and how it's quality controlled. So, for example, you can buy chitosan from Sigma, but they really don't give you enough detail to be able to parse any of this out. Whereas if you find a supplier that's already figured out how to make pharmaceutical grade batches of this, you're going to get much more reliable, QC data from them. And a lot of these suppliers do sell more affordable scientific grade stuff that hopefully is a little bit more one-to-one. But yeah, if you're looking to translate into an actual clinical product, then identifying those suppliers early will make things easier. And then also, this has been eye-opening just in terms of understanding what assumptions I was making and what assumptions folks in the literature were making, especially with chitosan, which is such a sticky molecule. It really causes a lot of problems. So for example, if you're looking at cytokine assays, there's really convenient bead-based assays that can, look at these massive panels. Can't do that if chitosan's in your sample because it'll stick all the beads together and you can't read out any of your data. There is a lot of literature done in just plain monolayer cell culture, but how translatable is all that if our hypothesis of exfoliation means that it requires tight junctions between cells or an intact immune system, et cetera. So there, it's been very interesting, rewarding, and I would lie if it wasn't frustrating to try and design around all of these various issues, but it makes me feel good to know that I've identified all these potential problems and hopefully increasing the rigor of the science. So this is my last slide to summarize the career development aspect of the K12. And so one thing that I struggled with during this year was not feeling as productive as I was used to, just in terms of doing experiments. But it helped to give myself a bit of grace and recognize all the other less visible work that I was doing, which is planning out a 5 or 10-year research vision, understanding what I need to do that, ranging from how much money do I need to pay a postdoc salary until I can get my own funding? How much does a -80 freezer cost? Where am I going to put the freezer? And then strategizing how to negotiate for a startup package that will help me succeed in doing that. And now that I am looking to actually hire people, how do I be a good boss and set clear expectations? What are my expectations in running a lab? And so recently, I actually took a little bit of time to write up a lab manual, which felt a little silly at first, given that there's no lab yet, but it's on the way. But it was a really, I would say, like vulnerable exercise because it's really asking you to confront, like, okay, how what are your values? What are you going to prioritize? How are you going to mentor people? But I felt a lot better and a lot more clear-headed about my goals after doing it. So really helpful exercise, and I'd encourage other folks to do it at any stage of training, especially this one. And yeah, I'll end with acknowledgement. So thank you very much to Joe and Chris Prado as my primary mentors for this K12 project, but there are many, many other people who have supported me along the way.